Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, from today onwards, we are going to start learning about cancer biology, a field where we are going to learn about the cellular and the molecular basis of the disease known as cancer. So, all of us know that the uncontrollable proliferation of cells leads to the cancer. So, in this series of lectures, we are going to discuss about how a normal cell it divides uncontrollably and it leads to it gets transformed into the cancer cell. We are going to integrate our knowledge of the genetics, the biochemistry, immunology, and the cancer biology to understand deeply about the cancer as well as the tumor properties. Okay, so this is kind of an introductory lecture where we are going to brush up all of our ideas about cancer, the tumors, the type of tumors that we have learned till now from the high school to the undergrad level. Okay, so before discussing about that, I would like you to know about certain terms, such as those terms, because I will be using those terms repetitively during the course. Okay, so the first term is known as hyperplasia. Hyperplasia refers when the altered cells. So there is something which is altered in a normal cell, which results to the uncontrollable growth. So those altered cells divide and increase in number, but when looked under the microscope, they look normal. Okay, so when they are visualized under microscope, they look normal. So let's say this is a tissue, okay, and this is the array of normal cells. Okay, so those are normal cells that this is how they are supposed to look under the microscope normally, but there is some alteration in those normal cells and they undergo, so they show the excessive division. Okay, so let's say that they are dividing excessively, and that is why the number of cells is increased. So here you can see the number of cell is increased. So this is a hypothetical schematic, okay? So the number of cell is increased, but when you visualize that under the microscope, the cells, the new cells, so the new tumor cells, they look similar to the normal cells. So we can say that when there is increase in number of cells and those cells look normal under the microscope, this condition is known as hyperplasia. Okay, so there will be no visual differences from the tumor cells to the normal cells. That condition is known as hyperplasia. The dysplasia, unlike the hyperplasia, refers to the excessive division of the cells, which leads to certain abnormalities which can be visualized under the microscope. Okay, so you know that something goes wrong in the cell, so there is something abnormal in the cell, and that is leading them to uncontrollably divide. But that, if that is visualized under the microscope, we then we call it as dysplasia. But it cannot be. It leads to normal looking cell. We call it as hyperplasia. So when the cells they look abnormal, so the phenotype is abnormal. So could be immunophenotype, or the coloration could be abnormal, or size could be abnormal, or the cells could look disrupted cells. Okay, so such condition is known as dysplasia. So there the excessive growth or the excessive division of cells, which leads to the abnormalities in them, which is visualized under the microscope. So those cells are abnormal looking cells. So you can by looking under the microscope, you can clearly distinguish between the normal cells and those growing cells, the proliferating cells. Okay, so there's one more term known as anaplasia. This refers when the cell do not undergo the differentiation. So we can say that anaplasia means lack of cellular division. Sorry, cellular differentiation. So when the cells do not undergo differentiation, this condition is known as anaplasia. Okay, I hope those terms are clear to you all. Okay, so now we are going to learn about the tumors, the types of tumors. Okay, the categorization of the tumors based on whether what are the characteristics of them. The first categorization that I'm going to do, the benign and malignant tumors, that categorization will be based on the characteristics of cells which are forming the tumors. So, for example, in benign tumors, those are non-cancerous tumors, means they do not have the capacity to invade and spread throughout the body. So, we call them as non-cancerous. Okay, non-cancerous. I guess there's something wrong in the spelling. Non-cancerous, then that should be cancerous. So yeah. So benign tumors are non-cancerous, means they are non-invasive, means they do not have the capacity to metastasize. I hope you know about the metastasis. Metastasis means when the cells, the tumor cells, able to they are able to spread throughout the body. That is what we call metastasis. Okay, so they do not have the capacity to metastasize. Can't They can't metastasize, so we do not see the metastasis here. The other category of the tumors which has the opposite characteristics from what benign tumors have, that is known as malignant tumors. So malignant tumors are the cancerous tumors. Okay, so they lead to cancer. So they are invasive in nature, means they can invade they can invade and spread throughout the body. Okay, so they show the metastasis. They are capable of metastasizing. Okay, so these are the basic differences between benign tumors and malignant tumors that we already know. There's other characterization of the tumors based on from which cell type they are arising. Okay, so this was based on certain characteristics, whether they are invasive or not. There can be other categorization based on the cell type, okay, from which cell that tumor is arising. So there's one more classification. So this is classification type 1, and here is the classification type 2, and this is based on cell type from which the normal cell the normal cell that is going to be the cancerous cell, or let's say that is going to be the tumor cell now. Okay, so based on which cell type is converting to the tumor cell, we categorize them as follows. If the cell is the epithelial cell, okay, so epithelial cells are the cells which form the linings in our body. Such cancer is known as carcinoma. Okay, carcinoma is the cancer of epithelial cells. Again, there is categorization based on which type of epithelial cells are undergoing the transformation from the normal cell to tumor cell. If the cells that produce fluid or mucus, they are causing the cancer, they are converting to the tumor cell, we call them as adenocarcinomas. So adenocarcinomas, this means that the cells which are producing mucus or fluid, means the secretory epithelial cells, okay, they are undergoing the transformation. So this cancer is related to them. Okay, so the breast cells, the colon, all of them, they are covered in the adenocarcinomas. Okay, there's the type known as basal cell carcinoma, where the basal layer of the epidermis undergoes the transformation. Okay, this is related to the basal layer of the epidermis. So the basal layer of epidermis. Okay, there's other type known as squamous cell carcinoma. So we know that the squamous cells, they form the outer surface of the skin. So when there is uh, there is transformation from normal cell to the tumor cell, so the tumor is formed from the squamous cells. We call them as squamous cell carcinoma. And there is one more epithelial cell type known as transitional epithelium, which is found in our bladder, the kidneys. Okay, so the carcinoma, this cancer related to those cells, is known as transitional cell carcinoma. So I hope you know that transitional epithelium is also so this is not that different from normal epithelial cells. So they also form the lining, but they have somewhat stretchy capabilities. Okay, so they can be stretched, and that is why we can see them in urinary bladder and all. Okay, so this transitional cell carcinoma, it is related to urinary bladder, kidneys. Okay. So the excretory part that has the transitional epithelium that shows the transitional cell carcinoma. So carcinoma is related to the epithelial cells. So when the mesenchymal cells, the cells of the connective tissue, they undergo this transition. So the mesenchymal cell tumors or the cancers, they are known as sarcomas. Okay, so the blood vessels, the muscles, when they undergo the cancer, we call them as sarcoma. So the very famous example, the very commonly found example for this sarcoma is osteosarcoma. Osteo means whatever which is related to bone, we term it as osteo. So osteosarcoma is obviously related to bones. Okay, and this is very commonly found sarcoma. Okay, if the blood there is other if the hematopoietic cells means the cells which are blood forming the blood, okay, or the blood cells themselves, they undergo the cancer transition. So let's say that hematopoietic cells. So the hematopoietic cells, there should be E here. Hematopoietic cells, if they undergo the transition, so the blood forming cells undergo the transition, we have the different names depending on which cell is undergoing. So we call the leukemia when the blood forming cells, they are like the bone marrow cells, they are showing the so the tumor is formed in those cell types, we call them as leukemia. Okay, so the blood forming tissues like bone marrow, so there is so again in the leukemia there is characterization of acute leukemia and the chronic leukemia. Okay, acute myeloid lymphoma. Okay, sorry, acute myeloid leukemia and all. Okay, we will learn about that in detail in the specific lecture about leukemia. Okay, so there is the plasma cells, the cells which are the specialized B cells. If they undergo if they undergo this transition, we call that as myeloma. Okay, so the cancer which begins in plasma cells, the activated B cells or the differentiated B cells, we call that as the myeloma. Okay, or let's say multiple myeloma. So those myeloma cells, they are abnormal plasma cells, and when they start building up in the marrow, that is known as Kale disease. Okay, let's not discuss it about here. So myeloma, it is basically related to the plasma cells. Okay, and there's again lymphoma, which is related to the lymphatic organs, the lymph nodes. If they undergo the tumor formation, we call that as lymphoma. So the third type, again, it is under the hematopoietic one, known as lymphoma. Okay, in the lymphoma, the lymph organs undergo the this transition. So the types are Hodgkin lymphoma and non-Hodgkin lymphoma. We will discuss about them in detail in again the dedicated lecture to lymphoma. So the Hodgkin and non-Hodgkin lymphoma. Okay, so the characteristics is that Hodgkin lymphoma that can be developed in the B cells. Okay, and this non-Hodgkin lymphoma, it can be developed in B cells or T cells. Okay, so the Hodgkin lymphoma is characterized by the presence of certain cells known as Reed-Sternberg cells. So those Reed-Sternberg cells, they were identified by the scientists known as Reed and Sternberg. So they identified certain cells which were multinucleated and the size was large, and they had some unusual immunophenotype, and this is the characteristic of Hodgkin lymphoma. So Hodgkin lymphoma, they have Reed Sternberg cells. cells. So those are large multinucleated. These are large multinucleated cells which has unusual immunophenotype. So immunophenotype means you can visualize that under the microscope. So you can see that those cells are somewhat different from the normal cells, and that is why they are known as Reed-Sternberg cells. And the presence of Reed-Sternberg cells signifies for the Hodgkin lymphoma, and non-Hodgkin lymphoma does not have those cells. Okay. So this is about hematopoietic hematopoietic tumors. Okay. There's one more type if it is related to the nervous system, known as neuroectodermal. If the neuroectodermal cells undergo the cancerous transformation, the nervous system cells, they depending on which cell type, specifically which cell, which glial cell or which cell is undergoing the transition, we call them with the different names such as gliomas, astrocytomas, then neurofibromas, meningiomas. Okay. So so this is brain and spinal cord tumors. Okay, when the spinal cord itself it shows the tumor, which we call them as intramedullary tumors. Okay, the spinal cord itself has the tumors, but if the membrane of the spinal cords or the nerve buttons that nerves that originate from the spinal cord, they are showing the tumors, we call them as extramedullary tumors. Okay, so the extramedullary tumors, they are seen in the membrane of spinal cord. So not the spinal cord itself, but the membrane of spinal cord and the nerves that are arising from the spinal cords. They show the tumors, then we call them as extramedullary tumors. The intramedullary tumors are in the spinal cord itself. So the examples are gliomas, glial cell undergoes that, that transition. Then astrocytoma, the astrocyte cells undergo the transition. Meningiomas, the brain coverings. Okay, neurofibromas. So these are the names. Okay, let me list them down. Gliomas, the astrocytomas, meningiomas, and neurofibromas. Okay, so this is how we classify the tumors based on from which cell type they are arising. So okay, again the treatment of the neuroectodermal tumors, that is quite tricky because you might know about blood brain barrier. Okay, so this barrier protects our brain from, and it does not allow the entry or passage of certain molecules. So that is why the treatment treatment becomes difficult, because whatever we are introducing in the patient, that would not pass the blood brain barrier if it exceeds the certain weight. If it exceeds certain kilodalton size, they could not pass through the blood brain barrier, and that is why treating the neuroectodermal tumors gets tricky. And that is why we say that is why this is generally a lethal condition. Okay, so so this is about the classification of the tumors. Okay, but due to our misinterpretation or misdiagnosis or the lack of the screening or diagnostic tools, a lot of times there gets the situation gets confusing between benign and malignant tumors. Okay, so this is rather often seen in the ovarian carcinoma. Okay, so ovarian cyst, so this is a benign tumor. Okay, so this is the benign tumor, and while the ovarian carcinoma, This is obviously the cancer which is related to ovaries. So ovarian cancer carcinoma is related to ovaries as well as the fallopian tube. I hope you know about the fallopian tube. It connects the uterus as well and the ovaries. So the fallopian tube is the site for the fertilization. Okay, so in the fallopian tube and in the ovaries, if the tumors are seen which are cancerous, which can spread throughout the body, we call them as ovarian carcinoma. So this ovarian carcinoma or the ovarian cancer that has high death rate due to the misinterpretation. We do not have the proper diagnostic tools to differentiate between both of those. So misdiagnosis often happens. Like in certain cases, there could be false positive cases or there could be false negative cases. So for example, if a patient has ovarian cyst, it could be diagnosed as ovarian carcinoma, or even if the patient has ovarian carcinoma due to no proper screening test, we might conclude that it is a cyst, and that can lead to, and that is why this cancer has high death rate. So the diagnostic tools for the cancer here, they are the imaging tests such as ultrasound can be done, and the CA25, CA125, the cancer. This is cancer antigen. Okay, this CA25 125 blood test, CA125 blood test is done to recognize. So this is the blood test that is done to identify whether the patient has the cyst or the carcinoma. So there is a basal expression of this antigen that is seen in all of the individuals. So there is a normal value that should be obtained in the blood, but the elevated levels of this antigen that indicates the cancer. So depending on the further surgical evaluations such as biopsy and all, we conclude whether the patient have the ovarian cyst or carcinoma. So this is the first case where due to misinterpretation, there is high death rate. Okay, and that is because we do not have the proper diagnostic tools, and a lot of times there is misinterpretation. The other case, such as this, is the BPH, the benign prostate hyperplasia. So this is a benign condition, benign prostate hyperplasia. So you know now that what hyperplasia means. Okay. So this is the benign tumor in the which is seen in prostate. Okay, and obviously the prostate cancer. So here also there is due to the difficulty in screening methods. So we do not have defined screening methods. So that is why there could be a confusion between whether the patient have VPH or prostate cancer. So the imaging techniques such as MRI and ultrasound, they are used for identification for the diagnosis. And there is a blood test known as PSA. So this is prostate specific antigen blood test. So all of us, the normal patient, all of us, the normal individuals have a certain level of the PSA expression. The elevated level suggests that it could be the hyperplasia or it could be the cancer. So the PSA blood test, prostate specific antigen blood test, it is helpful for this for the diagnosis of the cancer or whatever benign tumor. Okay, so again so there are certain cases like this. So other than this, there is osteochondroma and osteosarcoma. The osteochondroma refers to the pain and swelling in bones. So this is the benign benign tumor, and the osteosarcoma, obviously the cancer related to bones. So that can be identified by X-ray, MRI, and obviously biopsy. So there are multiple cases where the benign condition could be misinterpreted, or malignant condition could be misinterpreted, and due to improper diagnosis or the poor diagnostic tools, there could be confusion. There could be cases such as false positive and false negative, which could lead to high death rate. So we are still developing, the research is still going on on the proper diagnostic tools for for avoiding those issues. Okay, so in a lot of countries, in the developed countries, there are certain tests which has an array. So for example, they will be doing the multiple multiple antigen test, and they will be testing the urine, blood, and a lot of things, a lot of research is going on, so that not so that to avoid those problems, we can seek for multiple diagnostic tests, like multiple screening tests. Instead of doing only the CA 125 blood test or PSA blood test, we can do a lot of tests, like we can do the ultrasound plus the CA 125 blood test plus the other antigens. We can also test the urine, so that we will have the less chances of having the false positive and false negative cases, and the diagnosis could so the chances of diagnostic being correct, that can be increased, and that can lead to the saving a life. So that's all in this lecture. From in the today's lecture, we covered all of the basics about cancer. From tomorrow, we will learn about in detail about those cancer types. Till then, see you. Bye bye.